Hello students, today we will start with new topic, that is Mendelism. So what is meant by Mendelism? The contributions given by Mendel is called as Mendelism. Mendelism is considered as the basics for modern genetics, and whatever the work that was done by Mendel during his lifetime, it is considered as it is published with a name called as Annual Proceedings with a Natural History Society. This is a journal. Okay, this is a journal in the year 1866. And whatever the experiments are there that was done by Mendel, they are published with a name called as Experiments of Plant Hybridization. Mendel is considered as the first systemic researcher. What is meant by systemic researcher? Systemic researcher means they used to collect the sample, they used to frame out the ideas, they used to do the experiment repeatedly number of times and give very exact, clear, analytical details and data about so and so work done by them. Okay, their work will be very much systematically done. Term Mendelism was given by Karl Korenz. Mendel is first systematic researcher. Systemic researcher. First systemic researcher. Now, Mendel worked approximately on 24,000 approximately 24,000 number of pea plants. Mendel worked on approximately 24,034 number of pea plants. And for how many years did Mendel conducted his work on pea plants? Mendel did work, or simply we can say Mendel worked for 7 years on pea plants. That is between 1856 to 1863 years. And scientific name of pea plant is on Pisum sativum. Mendel died in 1884. Next, after Mendel's death in the year 1900, three number of scientists got some basic details about whatever that is work done by Mendel, and they started further work and research on Mendelism. Those three scientists are after Mendel's death. In 1900 year, three scientists. What is name of those scientists? Named Hugo de Vries. Next, Karl Korenz and Schermark. These three scientists worked on Mendel's work and his contributions. Now, Hugo de Vries belonged to Holland. What is necessity of studying about their location? This is because by mistake, if in the match the following, if we get such type of matches or questions, then we must be able to answer. Now, Korenz, he belongs to Germany. Schermark belongs to Austria. Next, Mendel was a mathematician, but he is not a biologist. We can also say Mendel is a mathematician and a naturalist, but not a biologist. Then what is meant by the difference between the naturalist and biologist? Let's see some of the points. A biologist can do the collection of the samples, and if he get any new ideas, he can bring the collected sample to his lab, and he can do the experiment in his lab along with the work done in the specimen available area. But a naturalist will do work only outside the lab. He is very much in contact with the natural work, and also, for example, if you get a specimen in the nature, means outside the lab, mostly his work will be outside the lab only. Such type of work and such type of name is given for those scientists as they are naturalists or are they biologists. Mendel was a mathematician and a naturalist, but not a biologist. Means he did not do his work inside the laboratory. Naturalists will do most research in nature only, not inside labs. So that is the difference between a biologist and a naturalist. Mendel studied in University of Vienna and he worked as a priest in a church. In a church. Now, we should not use the term like worked. Mendel was assigned. Mendel was assigned as a priest in a church. Should not use the term like worked. Now, reasons why Mendel's work was not recognized during his lifetime. In those days, publications are considered as quite normal and not much advanced. So, basing on that not much advanced and consideration as normal things, Mendel's work was not that much recognized in those days. Now, we will see reasons why Mendel's work was not recognized. First point, in those days, publications are very normal and not much advanced. So, these publications remained unrecognized for many years. As Mendel was a mathematician, his work included statistical analysis and data analysis. Okay, that we will write in the form of a sentence like, since Mendel was a mathematician, he included statistical methods and also analytical data which biologists did not understand. This is also one of the reasons why Mendel's work was not recognized in those days. As Mendel was a mathematician, he included statistical methods and we can use a term like mathematical logics. Mathematical logics, which biologists did not understand. This can also be one of the reasons why Mendel's work was not recognized. Now, we can also take another important thing. Mendel, during his work, he used a term called as factors. Now, we are calling it as genes. But if a proof was asked, he couldn't provide any proof for the existence of the factors that he used during explanation. So, it is also one of the reasons. That one we can write in the form of Mendel's used a term called as factor in which he couldn't provide the proof. Mendel talked about factors in which there was no proof. He couldn't provide the proof. This lacking of cannot providing proof might be because of lack of microscope. Could not provide the proof. Now, the factors term, factors are also called as discrete units. Discrete units. What is meant by discrete units? Discrete units means any of the genetic material. Now we are extracting some meanings from those, some technical terms. The separate forms of genetic material that can express themselves, though they are in homozygous form, heterozygous form, they are getting expressed separately. For example, when tallness, phenotypic genes, okay, dominant genes are mixed with recessive genes, only dominant genes are getting expressed. They are showing their own dominance. Okay, so discrete units means the genes or alleles that are expressed separately even in heterozygous form. Heterozygous form. For suppose, here we can take one example, a combination of alleles, one dominant allele and one recessive allele, considered as a gene in an organism. Now, this will be expressed since it is a dominant allele. It is responsible for the phenotype, that is tallness, and this is getting suppressed. Means this is getting exhibited its own special characteristic feature. This is not getting expressed in presence of dominant allele. We can also take some other example. For suppose, if two different types of genes are existed. For suppose, the genes that are responsible for round shape of the seed, if they are mixed with tallness of the plant, then the plant will be showing much height, and also its seeds will be round in shape. Seeds will not be in wrinkle. Means this is getting expressed separately, this is also getting expressed separately, but in the same plant. So this type of gene is responsible for a round shape of seed. This gene is responsible for height, tallness of plant. Means when two dominant genes are side by side also, both of them are getting expressed separately, but both of them are in same plant. Same plant body also we can use the term. Contrasting characters expressed by heterozygous alleles So these are also called as allelomorphs. Allelomorphs means the phenotypic nature exhibited by different types of alleles even at their allele forms. We know that genes are not giving rise to alleles. Genes are being expressed as allele forms or different types of forms that we are calling as alleles. Next, reasons why Mendel's work was recognized after his death. So what are the major contributions that are included in his work? So we will see about that also. Reasons why Mendel's work was recognized and why Mendel's contributions are successful in genetics field. First thing we can take that as Mendel has used approximately 24,000 number of pea plants, we can use a sentence like taking huge sample or taking or selection of huge sample. This can be one of the reason. Next, selection of contrasting characters. So, how many number of characters are selected by Mendel in the pea plant? How many number of contrasting characters are selected by Mendel in pea plant means characters selected by Mendel are 7 and contrasting characters selected by Mendel are 40. What are those? Yes, we will see after this topic. Next, what are the reasons, major reasons for his success in the field of genetics? Because Mendel is also considered as the father of genetics. Now, some more points. Mendel gave correct data analysis. And also, in the pea plant, there are no linkage of genes. I will explain what is meant by that sentence. No linkage of genes in pea plant. I will explain this one now. Very important one. As a mathematician, as a statistical analyst, what he has done, he maintained the records. Now we will see about this sentence. There is no linkage of genes. We have one special topic in this chapter about linkage of genes. We will see at that time in detail about those linkage groups and linkage of genes. Just I will give a brief idea what is meant by this sentence. We will see some details about this. No linkage of genes. Chromosomes, if they are like this and different forms of alleles are arranged in different positions on a chromosome. See the gap between two different alleles on a single chromosome vertically, here the gap is more. If it is crossed with its another homologous chromosome which is having the allele forms like this, now this one can easily pair with the neighboring or homologous chromosome and crossing over can happen. Now, in genetics, it was said that if the gap between what is meant by linkage of or linked genes are the types of allele forms or alleles which are present on the same chromosome vertically. These are also considered as linkage groups when the distance between the alleles vertically in a chromosome is very less, then they can easily bind with each other, and that is majorly leading for self-cross. But here crossing over has happened, and this can lead to cross-pollination or crossing over in case of animals. Crossing over of alleles. If the gene arrangement or allele arrangement is like this, now if genes are arranged like this, very nearby vertically on the same chromosome, then these are called as linked genes. Now here in such a condition, there is very less chance for crossing over. But according to Mendel's contributions, it was said that the alleles on the chromosomes of Poisson sativa, they are not like this, but they are away from each other. It means that the alleles or the genes are not linked. They are not nearby like this. If so, the crossing over cannot happen due to lack of the gap between the alleles, and crossing over cannot happen and cannot give the new variations or varieties in the Poisson sativa. So here, this is just one sample explanation between the linked genes and non-linked genes. Again, clearly I am telling, in case of Poisson sativa, allele arrangement is not like this, because if it is like this, that particular plant can do only self-pollination. And we know that Poisson sativum is giving different types of flowers like purple flower, white flower, etc. Contrasting characters morphologically are seen there. It means that the genes or the allele forms are away from each other on the same chromosome at different positions, so that crossing over has happened and we are getting different types of colors. Pea plant or Poisson sativum, it has 14 chromosomes. Now, name of character, chromosome number. It doesn't mean how many chromosomes, which numbered chromosome out of the 14 are responsible for so and so character. What is name of dominant phenotypic character? A recessive phenotypic character. Okay, so first, how many number of characters are studied by Mendel? Are 7. Are 7. Name of the character, if it is height. The chromosome that is responsible for the character height is chromosome number 4. Out of 14, the 4th chromosome, 4 or numbered chromosome, is majorly responsible for height of the plant. Now, phenotypic character is called as tall. Now, tall is represented genotypically with the alphabet capital T. Genotype. Expression is capital T. Now, actually, here we have to write 2 allele forms. So, we will write in a pair, allele pair, as 1 and 2 allele numbers. So, here it is dwarf phenotypically, which is expressed with genotype, allele pair, small t. In the same way, second type of a character is flower position. Flower position. This is also regulated by chromosome number 4. Here, dominant character flower position is axillary position. Axillary position of the flower. What is meant by axillary position? If this is a branch and if this is the leaf, then this area is called as axillary position of the flower. Axillary position. Recessive one is terminal or apical position. Apical position. Next, flower color. Flower color. Flower color, it is regulated by chromosome number 1 out of 14. Dominant one is purple. Recessive one color phenotypic is white. Now, fourth one, pod shape. Pod shape, it is regulated by 4th chromosome. And here the dominant one is inflated. It means that if this is the pod which is having seeds, the outermost layer is not having any bendings or constrictions. It is very normal and straight. Second one, constricted. Constricted. See. Next, fifth one. Fifth one is pod color. Pod color, it is also decided by chromosome number 5. And here color, dominant color is green. Recessive one is yellow. Here, next one. Seed shape. It is decided by chromosome number 7 out of 14. And seed shape, dominant one is a round shape of the seed. We can also use a term like seed cotyledon. Seed shape will write simply because finally cotyledon shape is deciding the seed shape. Here it is wrinkled. The last one character is seed cotyledon color. Now, it is decided by chromosome number 1. Chromosome 1. Now, seed color. Here dominant one is yellow. It is reverse of pod color. And it is recessive one is green. Here we have seen this one is decided by capital A. And here it is small a, we can write. Purple color with capital P. Small color with small alphabet. Inflated we can write capital I. Constricted we can write small I. Green we can write capital G. Here we can write small g. Round we can write capital R. Small wrinkled we can write with small r. Yellow with capital Y and green with small y. We can write different arrangements like this. Because of that only we tell characters are 7 and contrasting characters or different alleles that are responsible for 7 characters are 14. Even variations are also considered as 14. So 1, 2, 3, 4, 5, 6, 7 characters. Characters and contrasting characters 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Here the gene, one type of gene, 2. Third type of gene, 4th one. Fifth type of gene, 6th one. 7th, 8th, 9th, 10th. Means the genotypic arrangement. They are also 14 in a number. Okay. So this is about the Mendelism, his contributions and what are the 7 characters selected by Mendel. In the next class we will see about Mendel's laws.